Hey, it's Cassandra Scott from Biology Lab. Here is the cheek DNA isolation kind of experiment. Here are my ingredients. I have the dishwasher soap already measured out 1.8 mLs. And then here's my table salt, my distilled water 8.5. Here's my lysis buffer, the table salts. This is for the mouth rinse, and here is for my Pro-T solution. All right, so starting off, we're gonna go ahead and start with the lysis buffer. So first off, we are going to add the dishwasher soap. So I'm adding 1.5 mLs of dishwasher soap. And that's in the lysis buff. And then here is my 8.5 mLs of distilled water. And then here's my iodinized table salt. And then we're gonna stir gently until the solution is combined. Alrighty, and now that's pretty well combined. We're gonna go ahead and make our Pro-T solution. So I'm gonna put this down, and then we're gonna come over here, and with a pipette, we're gonna measure out 10 mLs of distilled water. This is my cup of distilled water that I have set aside. So 10 mLs of distilled water into our little beaker. And then here is our 1/8 teaspoon of meat tenderizer. I don't actually have a 1/8 teaspoon, so we're just gonna half it. There we are. And then we're gonna stir this until it's combined. Perfect. And then for our next step, we're gonna go ahead and get our cheek cells. So to do that, you wanna chew on the side of your cheek for at least 30 seconds, and then kind of gurgle a little bit and spit the juice back into the cup. So there's our beautiful cheek cells. Awesome. And then we are going to transfer this to this pipette, the 15 ml pipette, very carefully as to not to spill it. Okay. And then we're going to take 2 mLs of the lysis buffer from here. So 2 mLs of the lysis buffer, and we're going to go ahead and add that to here. And then we're going to invert it 5 times. 1, 2, 3, 4, 5. And then we're going to set that upright. Let me grab a clean pipette. Alright, and then we're going to add 100 UI of the Pro-T solution. So there's 100. Perfect. Put the cap back on so it doesn't spill. And then we're gonna gently invert it 5 times. 2, 3, 4, 5. And now we're gonna let it sit for 10 minutes and we'll come back to it.